wherein the pollen tube is entering through the micropyle area, we call it as porogamy. Okay, micropyle is a pore-like structure, so porogamy. When it is entering through the chalazal area, meaning the basal side, so we'll call it as chalazogamy. And if it is entering through the integuments, you remember integuments?
Yes, ma'am.
Then we'll call it as mesogamy. So depending upon the area or depending upon from where the pollen grain or the pollen tube is entering the embryo sac, it can be porogamy, chalazogamy, and mesogamy. Now, we know that the filiform apparatus that is there in the synergid, it is responsible for guiding the pollen tube, isn't it? For helping the pollen tube to come inside. And I'll tell you one thing, that even though, for example, this is the stigma, the style, the ovary. Okay, inside the ovary, there is the embryo sac. Now here, what are these two cells called?
Ends or
The two at this side, they are known as? They have filiform apparatus. So they are known as?
filiform, sir?
No, they are synergids.
Synergens.
And then there is the egg cell, right?
Yeah, excellent.
And here, what are the three cells here?
These three cells are. Ma'am, actually, I haven't revised that portion.
Antipodal cells. Okay, and in the middle, there are the polar nuclei. Now, we saw that either the pollen tube will enter through the micropyle, it will enter through the chalazal side, or it will enter through the integuments. No matter whichever way it is entering, ultimately it will have to reach the synergids, so that, you know, it has like one of the male gamete has to fuse with the egg cell. Okay.
Yes.
So the filiform apparatus is there to guide the entry of the pollen tube. Now, after entering, like when the pollen tube enters, then one of the synergid, like if the pollen tube is entering one synergid, so after that, the pollen tube is releasing the two male gametes in the cytoplasm of the synergids. Okay? The two male gametes that are formed. Now, how are the two male gametes formed?
From two cell stage.
Two cell stage. The pollen grains are shed at two cell stage. Then what happens?
The vegetative cell divide.
Vegetative or the generative?
Generative cells divide.
Yeah.
To form
and form?
Two male gametes.
Correct. Two male gametes. Now, both of those two male gametes, they are released into the cytoplasm of the synergid. Right? Now, one of the male gamete would move and fuse with the egg cell. Okay? And this will form a zygote. Like normally, a male gamete and a female gamete, it fuses and ultimately forms a zygote. Right? This zygote will be diploid, isn't it? Why diploid?
Because there is a female gamete and a male gamete.
So what, like, okay, there is a female and male, then why is it diploid?
Because they both, because of fertilization.
What is happening in fertilization?
The fusion of male and female gamete.
Yeah, so how is the zygote, like after the fusion, how is it becoming 2N? Think, it's very easy.
Because of the fusion.
Yeah, that's true, because of the fusion, but how is it, like how is the fusion causing the zygote to be 2N? I'm asking a very basic question. Because the gametes are N.
Yeah.
Aren't the gametes haploid?
Yeah, because the gametes are haploid.
Yes,
so
that was the question.
Okay.
So after one of the male gamete fuses with the egg cell, it will form the diploid zygote. Later on, the other male gamete, the one that is still left, it will move forward and fuse with the two polar nuclei. Now see, in this process wherein the male gamete was fusing with the female gamete and forming the zygote, this process is called as syngamy. Syn means fusion. Gamy is gametes. So the fusion of gametes is syngamy. Now see this. So the other male gamete moves towards the two polar nuclei. Remember where is the polar nuclei?
Yes.
So it will move towards the two polar nuclei and then fuse with it to form the primary endosperm nucleus. And that primary endosperm nucleus is triploid. Triploid means?
3 gametes.
3?
Gametes. Oh no, 3 cells are present.
3 sets of chromosome.
Yes.
Always remember, diploid means 2 sets of chromosome, haploid means 1 set of chromosome, and triploid means 3. So here I am saying that after the second male gamete is fusing with the polar nuclei, it is forming a triploid structure. Triploid means 3N, isn't it?
Yes.
Now tell me how.
Because there are 2 male cell chromosomes, set of chromosomes.
No,
And
only see here the other male gamete, meaning only 1 male gamete is there. Then
also,
why is it becoming triploid?
there are 2 polar nuclei.
Because it is fusing with 2 polar nuclei, and ultimately n plus n plus n gives us 3n, right? So
Yes.
this process wherein the other male gamete fuses with the 2 polar nuclei, we call it as triple fusion. Okay?
Yes.
Because three things are being fused, right?
Yes.
And polar nuclei, what happens? They are earlier present like this. After a little while, they both combine and form one. So they become 2n. Okay?
Okay.
And normally the male gamete is n. So n plus 2n gives us 3n. Or you can say n plus n plus n is also 3n. But more of like you would say this one. Okay? Because the two polar nuclei that were originally there, it will fuse and form one nuclei. One nucleus. That is diploid. And then it will you can say fuse with the male gamete and form the triploid primary endosperm nucleus. Clear?
Yes.
Now, all in all, we are seeing that there are 2 types of fusions that are happening. The first one is syngamy, where the zygote is being formed. And the second one is triple fusion, where the primary endosperm is being formed. And because 2 types of fusions are happening at the same time in the same embryo sac, so we call it as double fertilization. So many a times, a lot of questions can be asked on double fertilization, like what is double fertilization or what are the parts of double fertilization. So you'd say syngamy and triple fusion. Got it?
Yes, ma'am.
So syngamy plus triple fusion is equal to double fertilization. Clear?
Yes.
So this is what? What do you think? Which kind of event this is? Is it pre-fertilization, post-fertilization, or fertilization?
Fertilization.
Fertilization. Very good. All right. So tell me quickly, what is the last thing you've written? See, and this thing also, number of chromosomes is what we call as ploidy. When we talk about N, we say it is monoploid. Mono means one, right?
One.
And
Yes.
it means one set of chromosome. This thing, you know, this is like how we learn ABCD and it is
Yes.
always in our mind. This is
Yes.
ABCD of bio.
Okay.
Anywhere you go, you will find this. So don't ever forget it, okay? Try to set it like ABCD and 1, 2, 3. So
Okay.
2 sets of chromosome is called as diploid.
Okay.
And lastly, there is 3 sets. Clear? Same way, if I write 4n, then what would it mean?
No?
Quadruploid, right?
Yes.
And how many sets of chromosomes would that have?
4 sets of
4 sets. You are understanding the word set?
Yes, yes. Set has
Right.
23 chromosomes in
No,
humans.
that is, yeah, very good.
Yeah.
23 pairs. So pair means 23 plus
23.
23. Very good. Okay. Tell me, what is the last thing you've written?
Pollen pistil interaction.
The thing is completed?
Yes, this is the result of continuous chemical dialogue.
Oh. Okay, now you can note it down.
Done, ma'am. Done, ma'am. Done, ma'am. Done, ma'am.
This part is done, right?
Yes,
Or
ma'am.
no?
Yes, ma'am. Ma'am, a little more. Yes. Done, ma'am.
Okay, any doubts?
No, ma'am.
All right, very good. Now moving forward, let us look at what happens after the fertilization has been done. Now, the post-fertilization events and structures or whatever things that are associated with the post-fertilization and whatever happens. Now, the first thing that will happen is the development of endosperm. So, right after the fertilization process has been completed, endosperm will be developed. Then, embryo will be developed. The ovules which are were there, they will develop into the seed and the ovary will become the fruit. So these two, the last two are the thing that happens very later on. But first, endosperm development takes place, then embryo development takes place. Now, the first thing is endosperm development. Now, the primary endosperm nucleus, right, which is the triploid structure, that will convert into primary endosperm cell. Now, the cell will be formed around that nuclei. Okay. Now, this primary endosperm cell, it divides repeatedly and forms the endosperm tissue. It will still be triploid only. Now, the cells of this particular tissue, that is endosperm tissue, they are filled with reserve food material, meaning they have abundant of nutrients present in it, so that when the embryo development starts, so that embryo, since slowly it is developing, like from the zygote, embryo will be formed. So while it is developing, it requires a lot of nutrition, it requires a lot of food, minerals, vitamins. So that comes from this endosperm. So endosperm, if someone asks you what is an endosperm, you'd say it is a nutritive tissue. Nutritive, it provides nutrition. Okay. Now, the primary endosperm nucleus, it undergoes successive nuclear division to give rise to free nuclei. Now, what happens is that the endosperm, they can be of two types: free nuclear endosperm and the cellular endosperm. Now see what happens. You know that in normal condition, whenever the nucleus will divide, it will be followed by the division of the cell. That is how it will result into two daughter cells, isn't it? We know
Yes.
that every nucleus gets surrounded by a wall. Remember how we were seeing the organization in embryo sac?
Yes.
But there is a unique phenomena wherein the nucleus divides, but no cell wall formation takes place. Okay, and it keeps on dividing. So ultimately, we have so many different nuclei, but this looks like I have drawn tail, but it's like so many nuclei in one cell without any kind of cell wall. So you will say that this one cell is multinucleated, isn't it? This is uninucleated. This is multinucleated.
Yes.
This condition is also known as syncytium, this same condition. Okay.
Okay.
Now, the primary endosperm nucleus, it undergoes successive nuclear division, meaning the nucleus divides, right? And ultimately it forms free nuclei, many nuclei. This stage, when no wall is present in between the nuclei, we call it as free nuclear endosperm. But when the walls are laid down, see how? Now we have separate cells. So this is cellular endosperm. Cellular refers to cell production or the formation of cell. Afterwards, the cell wall formation takes place, and now the endosperm will become cellular endosperm. Okay. Now, have you ever had coconut water?
Yes.
Hmm. And have you ever had that white portion that is to
Yes.
eat? Okay.
Yes.
See, the coconut water that we are drinking, it is also the endosperm, and the white, what is it called? I don't know.
Malai.
Yeah, yeah. Like, what is it called in English?
I don't know.
Even I don't know. So that
Uh,
malai of the coconut, that is the cellular endosperm.
okay.
Okay, meaning
Okay.
it was initially multinucleated condition, but then cell wall formation took place. That is why it becomes hard.
Okay.
But the water is normal like water only because it has no cell wall. Clear?
Yes.
Now, there are certain seeds in which the endosperm will be consumed by the developing embryo. See, endosperm is there so that whenever the embryo develops, so the embryo will get nutrition. That is the only reason it's there. Okay, we eat it, that's a different thing. But endosperm in any of the seed or plant or any of the food product, meaning derived from the plant, it is there to nourish the embryo. So if like sometimes it can happen that if the embryo is growing, it can take up all the nutrition and grow, right? So in such cases, the endosperm gets completely consumed. And that happens in the case of pea and groundnut. Like their endosperm is consumed by their own embryo.
Okay.
Then in the seeds such as castor and coconut, the endosperm is present. It has not been consumed. Okay.
Okay.
So endosperm development is clear.
Yes, ma'am.
Okay, very good. We are almost near to the end of the chapter.
Yes.
Coming on to the embryo development. Now let us see what embryo does and how is it. Now see, embryo development, it will obviously start at the micropyle end because the zygote is formed over there only, right? Because that
Yes.
egg cell was present on the micropyle end. So embryo develops at the micropyle end of the embryo sac where the zygote is situated. Now, most of the zygote, they divide only after some endosperm has made, right? Because they are
Yes.
like, okay, we will start growing when our food is there. If the food will not be there, we'll die. So
Yes.
they start dividing when endosperm has been formed so that the developing embryo gets the nourishment. The early embryo development is called as embryogeny. Okay, embryo is embryo, geny refers to genesis. Okay, genesis means formation.
Oh,
And this development of embryo, the pattern of development of embryo or the way or the steps that are involved, it is same for monocotyledons and dicotyledons. Monocots and dicots are the two types of angiosperms, and
yes.
in both of them the embryo development is more or less the same. Let us see what are the stages of the embryo development. Initially there is a pro-embryo, then from the proembryo, the globular embryo develops. Then from that, the heart-shaped embryo develops. And lastly, there is the mature embryo. So these are the stages, and these stages are common for monocots and dicots. Now talking about the dicot embryo, but before, and see in your book, there is a part where they have shown you 4 pictures like of these stages of embryo development. See in your book,
Okay. I'll check it
there's a
out.
diagram. Check.
Yes, ma'am.
Hmm. There, right? All these stages are there. So that
Yes.
is how it like it goes from one stage to another, and then that happens. Any doubts up until now?
No, ma'am.
Okay. Okay, now you can note it down.
Done, ma'am. Done, ma'am. Done, ma'am. Done, ma'am. Done, ma'am. Done, ma'am. Done, ma'am.